Alright guys, so this video is going to go through the remaining analysis questions which have to do with interacting proteins as well as where your protein is being expressed in the cell, and then everyone's favorite analysis question about future experimentation. So in order to figure out if any other proteins interact with your protein, we need to find a homolog in the Arabidopsis genome, and this is because the proteins of the Arabidopsis plant have been really well studied, while the proteins of the Landoltia plant have not been as well studied, hence we're helping contribute to the data pool that exists. So the first thing you need to do is you need to do a blast search for an Arabidopsis protein. And you can do this inside the TARE database, which is a database that is all about Arabidopsis. So there's a link in your DSAP question. If you go there, click on Tools, BLAST is an option, okay? And what you're going to do is you're going to do a kind of BLAST P option. So you want to blast the amino acids. So I need to grab my amino acid sequence here. And I'm going to paste it in. You don't have to change anything else. And then you can run the blast, hopefully finding a match in Arabidopsis. Then what we're going to do is we're going to take that match from our Arabidopsis plant. Okay, so here's the list of matches. I would ideally say start with the first match. So DSAP's probably going to ask you for the accession number and the name of the protein. So this is AT1G70410.2, and it is a beta carbonic anhydrase, which I think, if I remember correctly, is what our protein is in our Landoltia. We're going to copy this accession number, and we're going to paste that accession number into our interactome database. Okay, and basically what this database shows us is all the protein interactions, or all the known protein interactions, that exist inside an Arabidopsis cell. Okay, and of course, this is a giant map. There are lots of proteins that exist, so instead of just like diving into this haystack here, we're going to search our match, our Arabidopsis homolog, and very often this database does not like decimal points, and they're just kind of variants of the protein, so it's okay to get rid of that, and then hit go. If your Arabidopsis homolog interacts with any other proteins, you'll get a map like this. So here's my Arabidopsis homolog, so this is basically my protein that I have coded, and it seems to interact with itself, as well as 1, 2, 3, 4, 5, 6, 7 other proteins. DSAP's going to ask you about one of these proteins, so if you click on it, over in the margin you'll get some information about that protein, including the description, which is what DSAP's going to want to know a little bit about. Okay, so once you have your interacting information, you're going to venture to another database, which is going to tell you about where that Arabidopsis homolog is found in a cell. So I'm going to paste in my number. Again, I'm going to get rid of those decimal points. I'm going to place it in primary gene, and I'm not going to worry about any of this other stuff here. And I'm going to hit go. And what I'm going to first see is where in the plant, where in the Landoltia plant is this protein being expressed. And you could tell from the key over here that higher expression is shown by a red color, low expression is shown by a yellow color. So I'm seeing a lot of expression in the leaf in all of its phases. I'm seeing a lot of expression in the cotyledons, which are essentially early leaves that germinate from the plant. Okay, and I'm seeing a little bit in this first node of the stem, but those are my most noteworthy areas, it seems like. Okay, Then you might want to ask yourself, okay, this is great. This is where it is inside the plant. But if I go into a leaf cell, where in the leaf is it? Okay, so now we want to change our data source, and we're not so concerned with developmental map, but we're more so concerned with cellular location. And let me see if I can find it. Subcellular localization is what we want. And here we go, we're getting a view inside a single cell, okay? And same color key applies, max being red, min being yellow. And I'm seeing a decent amount here, probably the most, in the plasma membrane. Okay, so that's what you would record in your DSAP. Okay. Last question is going to be asking you about all those lovely experiments you might do. So again, I want to redirect you to the wssp.rutgers.edu page. Go to the resources tab. There are resources for students. If you scroll down, there is a molecular biology experiments, what to do next. There's a PDF you could read. There's a video you could watch. There's a PowerPoint you could view. Okay, all these things to help you determine what experiment you should do next. Make sure when you are writing this up, you are as specific as possible. Do not be vague. Do not be general. Do not say, I'll knock out my gene and see what happens. Think about what your gene codes for. What could happen? Okay? Think about the information you got from the TARE database and the interactome and the development chart. Okay? What might this help you want to figure out? What might you want to confirm? Okay? So think through this. Take your time on this one because this is the one that John Brick is going to be a stickler about. All right, guys. Best of luck. Keep analyzing. Always available on email if you need.